किधर आएगी यार, तुम कहाँ?
Ça c'est stand. Super. Ça, et c'est censé filmer. Ah, ça enregistre pas? So it's registered?
No, record. This is video recording.
Hope she will stand in the right place.
Yeah, she will keep
Maybe
moving.
you should stay. No?
Yeah. I also need to help with the other things, but it's okay.
Pleasure and the honor to chair this PhD course. So Garima Joshi today to defend PhD thesis. I'm trying on multifaceted anti-viral to capsid protein modulates PDC immune response. So you have 30-40 minutes to present your results, and then we will discuss the results. Thank you very much. So good afternoon, everyone. Thanks for being present here for my final thesis defense. So during my PhD, I tried to study the hepatitis E virus and the ORFg capsid protein and how it can modulate possibly the PDC immune response. So let's first talk about why I wanted to study the hepatitis E virus. It's a very dangerous virus. It's zoonotic and it has great risk of spillover to humans. And then there is a lack of a widely available vaccine against the hepatitis E virus. Of course, there is a vaccine, HEV, licensed in China since 2012. And recent US trials have shown promising results. But further studies need to be conducted. And finally, there is an overall lack of information about this virus. So it's the ignorance sometimes that kills. Sometimes there are not enough screenings of blood against the hepatitis E virus because there is not enough information about it. So the hepavirus genus was created quite recently, just 20 years ago in 2004. And it's the plasmavirus genus that causes pathogenesis in humans the most. Recently, it was discovered that the Roca hepavirus or the rat hepatitis virus was also spilling over to humans. But we will talk here mostly about the orthohepavirus A or the plasmavirus, which causes pathogenesis mostly in humans. It consists of 8 genotypes, 5 of which are pathogenic to humans. The hepatitis E1 and 2 is found only in humans. However, the hepatitis E virus 3, 4, and 7 are zoonotic and can be found in humans and other animals. Geographically, hepatitis E virus 1 and 2 are more localized in the developing countries, and hepatitis E virus 3, 4, and 7 are localized in the developed world. The transmission of hepatitis E virus 1 and 2 is mostly by contaminated water or food. Whereas for the 3, 4, and 7, it's usually uncooked meat, contaminated food products, blood products, and milk. Well, hepatitis E virus 1 and 2 are less likely to cause chronic infection, whereas the 3, 4, and 7 cause more the chronic infection. So today we will talk about the hepatitis E virus genotype 3, which I studied during my PhD. And this is quite prevalent in France, causing around 68,000 infections annually, and this number is around 300,000 in Germany. The transmission is mainly by uncooked meat, and so I just want to mention here that I brought some saucisson, but if you have certain prevailing diseases, don't take it. Things like if you have pre-existing liver disease conditions, if you're a solid organ transplant recipient, or if you are an immunosuppressed individual. These people are more likely to develop a chronic infection upon HEV infection. That's why immunity plays a very important role in the pathogenesis of this virus. And that's why we wanted to study it. Now moving on to the genome of the hepatitis E virus, it consists of a positive sense single-stranded RNA genome around 7.2 kb in length. And then a smaller subgenomic RNA can also be transcribed in the replication cycle of this virus. So the longer RNA strand, it gives rise to the ORF1 protein, which is a non-structural protein. And the subgenomic RNA or the shorter RNA can give rise to the ORF2 or the ORF3 structural proteins. So let's talk about the ORF2 and ORF3 a bit. So ORF2 forms the capsid protein, the capsid layer of the virus, whereas the ORF3 protein is associated with the envelope of the virus, and it helps in the release of the virus from the infected cell. And something really special about this virus is that it can exist in two forms. One is the naked virion particle right here, and the enveloped particle. So, going through quickly the replication cycle of the HEV, both the naked and the enveloped HEV can find their way inside the host cell and uncode, giving rise to an exposed RNA, which then translates into ORF1. ORF1 consists of the RNA-dependent RNA polymerase, which can then transcribe the negative sense RNA strand or the template strand that gives rise to either the full-length RNA of the virus or a subgenomic RNA, like I said before, which is then translated into the ORF2 and ORF3. This then leads to the assembly of the virus, which consists of 3 steps. So one is the decamer formation of the capsid protein. Second is the assembly of the decamers with the RNA. And finally, the formation of the complete virus particle, the icosahedral shape. And then these viruses can be released by the exosomal pathway. So I've been talking a bit about immunity, and it would be incomplete to talk about immunity without mentioning interferons. Interferons were first discovered in 1957 for their ability to interfere with viral replication. So here we will just focus on interferon 1 and 3, which have been found to be important in hepatitis E virus infection. So in terms of the location of receptor expression, the type 3 interferon receptors are more localized at the mucosal surfaces like the lungs, the gut, the reproductive tract. Whereas for the type 1 interferons, they are more ubiquitous in the body. For the kinetics of response, the response by interferon type 3 is more sustained and slow. However, the interferon type 1 is faster, it acts like a pulse, it rises quickly and goes down quickly. Now in terms of potency, definitely type 1 interferon are more potent than the type 3 interferon, but this does not come without a limitation. So there is a limitation of high potency of type 1 interferon, that is inflammation. type 1 interferons are more inflammatory than the type 3 interferons. Well, just to let you quickly know that inside the cell, the RNA viruses can be sensed by the VIR-I and the LFA-I, and the TLR3 and the TLR7-8 can also identify the virus by the in the endosomes. Now, the HEV has found various ways, various mechanisms to kind of inhibit interferon responses in the cells. For example, ORF1 here, it prevents the phosphorylation of STAT and translocation into the nucleus. ORF2, on the other hand, it prevents the phosphorylation of IL-3 and its nuclear translocation. The HEV ORF3 protein prevents the STAT phosphorylation. So, even though these mechanisms exist after infection with HEV, hepatic cells can produce interferon type 3, but no interferon type 1. Now, it has been shown by a lot of literature that hepatitis E virus can still persist despite the production of type 3 interferon. So, which means we should bring in the type 1 interferon here, because type 3 interferon definitely seems inefficient in controlling the virus. So, is there any cell type in the body that could be producing type 1 interferon? Well, there is no cell better than the PDCs, which produces 200 to 1,000 times more type 1 interferon than any other cell upon viral infection. And PDCs show a high degree of resistance to viral infections. And thanks to this phenomena, it cannot be infected by the virus. Therefore, the inhibitory mechanisms that the virus has against the different immune signaling pathways cannot be activated in these PDCs. So, by what mechanism, what can the interferon released by the PDCs do? Well, it can promote intrinsic antiviral immunity by the induction of ISGs, and also it has few immunoregulatory effects. It kind of acts as a bridge between the innate immunity and the adaptive immunity. So, it can promote cytokine secretion to recruit innate and adaptive immune cells, and also it can promote the effective functions of T cells, B cells, and NK cells. Now, there has been some literature that points out that an impaired interferon 1 response can lead to severe liver disease during acute HEV infection, which means if there is an impairment of interferon 1 response, a person is more likely to have severe HEV infection. And also that interferon 1 or interferon alpha monotherapy can inhibit HEV replication to some extent. So, the main question here is there a natural producer of interferon 1 in the context of HEV? And does the interferon type 3 production by hepatic cells make the HEV-infected cells impervious or resistant to interferon type 1? And these are the questions we really wanted to ask through our project. So that brings us to the first axis of my project, where we wanted to characterize the PDC response to HEV-infected cells. So for this, first we had to identify relevant cell types for co-culturing with the PDCs. And then we just wanted to see if PDCs have a response against the HEV-infected cells, and then to characterize the interaction between the PDC and infected cells if it existed, and then whether the interferon produced can actually control the viral replication. So first, to select the relevant cell types, we use different kind of inducers of immunity in the cells we chose. First, we trigger the TLR3 by just treatment with poly-IC in the supernatant. And then we also can transfect the poly-IC to trigger the REG-I and MDA5 pathway. And then we also can activate the paracrine signaling by treating with interferon lambda, TNF alpha, and interferon beta. So we chose HUH7.5 HEV2C3A cells and PLC3 cells, which are hepatoma cell lines, because they have already been well explored in the literature in the context of hepatitis E virus. So when we trigger the TLR3 pathway, we don't see an upregulation of different ISGs. So we are checking the ISGs here by QPCR. And so we saw that upon poly-IC transfection or REG-I-MDA5 activation, we saw an upregulation of ISGs only in HEV2 and C3A cells. There was some upregulation of ISGs after interferon beta and interferon lambda treatment. However, this was really low for TNF alpha. So in the end, we chose the HEV2 and the PLC3 cells for our experiments, co-culture experiment with the PDCs. But I should mention here that the PLC3 cells were already a well-established model by our collaborating team. So most of the results you will see here are done in PLC3 cells, where we also try to verify those results in HEV2 cells. So just to walk you through a basic co-culture experiment. So we have to prepare our cells before the co-culture experiment, around 6 days before, where we electroporate them with HEV RNA, and then we change the medium of the cells. And 6 days later, then we get a pouch of blood from which we isolate the PBMCs, and with positive selection, we isolate the PBCs. And then we put both the cell types in the same well, incubate them for around 18 hours, and harvest the supernatant to see what are the levels of cytokines. For us, we usually look at interferon alpha subtypes. So here we co-culture the PDC with infected cells, and you can see that there is some interferon response by the PDCs. Well, it's quite a lot compared to when we have no PDCs. So yes, PDCs can produce interferon response when they are co-cultured with infected cells. And then we wanted to see if the contact between the PDCs and the infected cells is important, as has been shown previously various times. So we introduce a barrier between the infected cells and the PDCs to see if the contact between the two cell types is important for PDC interferon production. And we see that when we introduce this barrier, there is a complete absence of PDC response, which means that the contact is really important for the PDCs to mount a response against the hepatitis E virus infected cells. So, canonically, PDCs can identify infected cells or the RNA by TLR7, which is present in the endosomal compartments of the PDCs. And this happens when the PDCs come in contact with the infected cells, and this is mediated by ICAM-I, which is on the side of the infected cells, and alpha-L-beta-2 integrin, which is on the side of the PDCs. Now we just wanted to verify if this was happening also in the context of the hepatitis E virus. And when we employed inhibitors against the TLR7, the IRSS661 inhibitor, we see that there is a complete diminution of response of the PDCs against the hepatitis E virus infected cells, which means TLR7 is really important for the PDCs to recognize the virus. And then when we employed inhibitors or antibodies against the alpha-L-beta-2 integrin or ICAM-I, we saw that there was a decrease in PDC response, but it was not as pronounced. Probably because there can be other mediators at the contact, we don't know yet. So what can the interferon response produced by the PDCs do? Well, it can do two things that can benefit the host. One is that it can upregulate the interferon stimulated genes. And two, it can control the virus and decrease the replication of the virus in the cells. So first, we just wanted to see without the PDCs, to what extent were the ISGs upregulated. And you can see in the HEV-2C3A cells, we see that almost all the genes were upregulated. The MXA, ISG15, TNF-alpha, IL-6, interferon lambda. However, in the PLC3 cells, we see there is an upregulation only of the interferon lambda and not rest of the ISGs. And then when we co-cultured with PDCs, we saw that for the HEV-G2, there is only an upregulation of the interferon lambda, and same for the PLC3 cells. So there is almost a global lack of ISG induction after 18 hours of co-culture of PDCs with infected cells. Now, how did that translate into the control of the viral replication? So we saw that, we did the same test in the two cell lines side by side, and you can see that the cell line which I showed was producing higher ISGs upon infection was able to control the virus more potently upon PDC co-culture. However, when you see the PLC3 cells which were not upregulating the ISGs so potently, you see that there is no difference in viral replication after 18 hours of co-culture, which means the initial ISG profile of the infected cells may be important even after the PDC has mounted an interferon response in the control of viral replication. Now, we wanted to see if the viral spread was also affected in the presence of PDC. So for this, we did flow cytometry because some wise person once told me that RNA is just not enough to see. You need to look at the protein level. So we had to see by fax if this was happening. And also, we wanted to also follow up for the PLC3 cells, which were not showing a decrease in replication at the 18-hour time point. We wanted to see if we extend this time point a little bit, will the replication be controlled or the viral spread will be controlled. So for this, we co-cultured the PDCs with the uninfected cells, which were also GFP positive, and the infected ORF2 positive GFP negative cells. So whatever new infection we were getting would be GFP positive cells, which also became ORF2 positive, which is a protein expressed by the HEV. So in the absence of PDCs, you see that there is this is just a reference, and when we have the PDC co-culture, there is a 50% decrease in the infection of initially infected cells. And when we co-cultured with the PDCs, even the newly infected cells were decreased by around 50%. So this means that at the 48-hour post-co-culture time point, there was a control of hepatitis E virus spread by around 50%. Now moving on to a more interesting aspect of the hepatitis E virus. What I showed you now previously has already been shown in previous articles, just not in the context of hepatitis E virus. Now we will discuss something really special to hepatitis E virus. Now I talked about the ORF2 capsid protein. Well, it's just not the capsid protein. The infected cells can produce ORF2 in glycosylated and cleaved forms in huge quantities. And there is some literature to point out that this ORF2 glycosylated and cleaved forms can probably bind neutralizing antibodies. So when the neutralizing bodies are engaged with the ORF2 glycosylated and cleaved proteins, they can no longer go and neutralize the virus itself. So they kind of act as an immune decoy for the virus. And then we have also a nuclear ORF2 that can shuttle in and out of the nucleus of the infected cell. So there are two main pathways of ORF2 production. The non-productive pathway by which the ORF2 glycosylated and cleaved forms are produced, and the productive pathway by which the ORF2 infectious particles are produced. And there exists a nuclear pathway by which the ORF2 goes in and out of the nucleus. Now, our collaborating team at Institut Pasteur Lille, which belongs to Laurence Coquerel right here, they have developed mutants for each forms of the ORF2. So I will just briefly describe the mutants of the ORF2. So in the hepatitis E virus wild type, we can see the ORF2 infectious form, the glycosylated and cleaved form, and the nuclear form. In the 5R5A mutant, the arginine-rich motif is mutated, so the ORF2 can no longer go into the nucleus. However, these cells are still producing the ORF2 glycosylated and cleaved forms. In the NES mutant, the nuclear export signal is mutated, so ORF2 does go to the nucleus, but it cannot come out of the nucleus, so there is a concentration of ORF2 into the nucleus. But other forms are formed as the wild type. And in the stop mutant, there is no glycosylated and cleaved form since the single peptide of the ORF2 is mutated. However, the infectious form and the nuclear form are still there. And then we had a replication deficient mutant, the GAD mutant. So this brings us to the second axis of the questions we wanted to ask. Do any of these ORF2 forms can modulate? Can they modulate PDC immune response? And we could do this thanks to the tools provided by Laurence Coquerel team by simply co-culturing the mutant infected cells with the PDCs. So we wanted to see if PDC response is modulated, and second, what are the causes of these differences. So just to walk you through quickly how these mutants have been characterized. So for the stop mutant, they introduced a stop codon in between the start codon of the ORF2 glycosylated and cleaved forms and the ORF2 infectious form. So the glycosylated cleaved form can no longer be produced, only the infectious form is produced. And they have verified this through different Western blot using different kinds of antibodies that stain differentially for the infectious form, the glycosylated and cleaved forms. And in the supernatant and within the cells, there is no difference in the viral RNA. And also, there is no difference in the infectious titers between the wild type and the stop mutant. Now, we had to do our due diligence. So we had to kind of characterize them microscopically, all these mutants. So we wanted to verify if the 5R5A, in this we did not have any nuclear translocation. Well, yes, indeed, we don't. And in the NES mutant, we had nuclear translocation a bit more than the wild type, and that's true. And then in the stop mutant, it's the distribution of ORF2 is kind of similar to the wild type. And this was same for both the HEV2 and the PLC3 cells. Now, first we wanted to just make sure that the replication of mutants versus the wild type was around the same by qPCR. And we saw that it was indeed the same. And when we co-cultured the PDCs with HEV2 cells that were infected with the mutants and the wild type, we saw that there was a slight decrease in the PDC response to the 5R5A mutant, in which the ORF2 does not go to the nucleus. That could possibly mean that the nuclear translocation of hepatitis E virus ORF2 or the ORF2 infectious form that the 5R5A is also not producing could somehow facilitate the PDC activation. And then we tried to see the same for the PLC3 cells. The replication of the wild type and the mutants was uniform for them. However, we saw a stark difference in the PDC response for the HEV2 cells versus the PLC3 cells. Here you can see a complete absence of PDC interferon response. And even the stop mutant is eliciting lesser PDC response. So this could probably hint at the role of ORF2 glycosylated cleaved form or the nuclear form that could probably contribute to PDC activation. And one of the hypotheses that we made was probably this was being regulated at the cell contact. Probably with the wild type, PDCs are making more contact as opposed to the mutants. So we tried to verify this, and for this we co-cultured the PDCs with infected cells and the wild type and the mutants. We incubated them for around 4 hours, fixed them, and then immunostained them for HEV ORF2. I just want to mention here that we also pre-stain the cells, so they are of specific colors. So the PDCs are blue, the PLC3 cells were red, and when we immunostain, we have green signal for the ORF2. And then, with the help of Jacques Broca, we set up a pipeline for the analysis of images that were obtained by microscopy. So here we first have the images, and we were able to identify all the cells, infected cells, and PDCs by the fluorophore they were expressing, or the signal, the color. And then we could select the number of synapses for the wild type versus the mutants. And there were 2 criteria for this. First, that the proximity of the PDCs and the infected cells was less than 1 micron, and that the area between the PDCs and the infected cells was more than 0 micron square. And then we put all the values that we got from this analysis into this formula, and we got this graph from that formula. On the left you see just verification of the infection percentage in the wild type versus the mutants that we always do. So we also do it here at the level of protein. And here you can see that for the wild type, the 5R5A and the NES, the percentage contact is around the same. However, this is decreased when the stop mutant is in contact with the PDCs. This is less than what is happening in the wild type. So probably the contact between PDCs and infected cells was diminished for the stop mutant. And it means that the glycosylated and cleaved forms that are being produced by this, that are not being produced by the PDCs, can somehow facilitate PDC immune response. That brings us to the first conclusion. Well, like I said, we chose HEV2 and the PLC3 cells as the most immunocompetent cells. And the PDCs produce interferon alpha robustly when in physical contact, and this contact is mediated by the ICAM-I and the alpha-L beta-2 integrin, and this RNA is sensed by the TLR7, which leads to the activation of the interferon signaling pathways in the PDC. And this interferon can lead to the decrease of infection or viral spread. Around 50% reduction is what we saw. And also I would like to highlight here that the immune status of the infected cells was really important in the impact of the PDCs on the viral control. So let's talk a bit about the mechanism that could be used by the ORF2 glycosylated and cleaved forms to facilitate PDC response. So the infected cells are producing glycosylated and cleaved forms, and this can go on and pre-stimulate the PDCs, perhaps, it's our hypothesis. And this can somehow facilitate PDC response or PDC by PDC contact. And something we did not answer yet, but we do seek to answer in our future experiments, is the question about how nuclear ORF2 can regulate the PDC response, because we see a huge difference in the 5R5A mutant and the wild type. So we think probably the nuclear ORF2 goes into the nucleus, somehow stimulates the interferon pathway, and it leads to the release of cytokines, which can then facilitate PDC contact or better screening by the PDCs. So for this, we would like to look within the cells. And what we'd like to see is whether the IRF3 localizes into the nucleus and whether this correlates with ORF2 nuclear localization. So as we know that upon viral recognition, IRF3 and NF-kappa B localize to the nucleus, and this can lead to the upregulation of surface molecules, which would mean more contact, and also release of cytokines, which would in turn facilitate more contact, better screening by the PDCs for the infected cells. So for this, we are trying to see by imaging flow cytometry. We can see at the single cell level the ORF2 nuclear translocation, whether it's translocating to the nucleus or not, or IRF3 nuclear translocation, since we apply a mask using the nuclear staining. So first we wanted to see the kinetics of ORF2 nuclear translocation. And here we see that the ORF2 localizes to the nucleus in the HEV-G2 cells starting 48-hour post-electroporation. And we see here we have two kinds of populations in the wild type. One population in which ORF2 goes to the nucleus, and another population in which ORF2 does not go to the nucleus. So you have these two populations here. And we see that when ORF2 does go to the nucleus, the IRF3 is more likely to localize to the nucleus, which means there might be a correlation between the IRF3 localization and ORF2 nuclear translocation. Now talking about the interesting directions that this project has opened up. So we still don't know what is the source of interferon lambda, whether it is the PDCs, the infected cells, or both the cell types, and whether the interferon lambda can give an advantage or is it disadvantageous for the interferon lambda to be produced by the infected cells. Since there have been some studies that show that the treatment of exogenous interferon is still not efficient enough in bringing down the viral replication. PDCs have acted in a more specific, more targeted manner. So maybe PDCs are still more efficient than the exogenous interferon 1 itself. And then we also saw that the PDC contact with the infected cells was not so drastically dependent on the ICAM-I and the alpha-L-beta to integrate. So maybe we can look at other candidates or do a dose-dependent inhibition of interferon response by using increasing concentration of antibodies. And then we can also profile the PDC surface molecules upon activation by HEV and compare them with other viruses. Also something we also tried to solve during my PhD was the PVC response when the cells are really infected with the virus. Like I told you, we electroporate the cells. We are not naturally infecting the cells with the virus because it's kind of a slow process. And you can imagine the PVC isolation itself being really slow. It would make us really slow, and I would finish my PhD in 8 years. So to just make the process a bit more rapid, we electroporated the cells. But we did collaborate with Julie Lucifora team and Loan Doughty team, who could infect the cells for us. But when we co-cultured these PVCs with these cells, we did not see the production of interferon at the protein level, at least. And then what is the mechanism by which the ORF2 glycosylated and HEV forms and the nuclear form could be regulating PVC response? It's still an open question, and we're still trying to answer it. In the end, I would like to take the time to thank my team, Marlon, Delphine, who have guided me, Laurence, who has provided us with very good tools, and Melvin and Laurence stepped in at a very critical time, which is also very important to us. Jacques, who helped me analyze all the images, and Antoine, who has helped me administratively in the recent times. I would also like to thank Alex Bello, Julie Lucifora, Soren Faludan, Elena Tomasello, who have helped me during my CST and given some really useful suggestions. And also I'd like to thank the BIMIC directors, Matthias Kor and Yuri Momo. And I would like to thank the jury. Thanks for your attention. I'll take any questions.
So now it's time to discuss. So we will start with your two rapporteurs who are online.